All right, we're gonna get started. We are gonna have today as our speaker Jingshu Wang. She graduated from Stanford back in 2016. Spent a few years at Harvard. No, sorry, at UPenn, and has been an assistant professor at U Chicago Stats Department since 2019. Her research interests are broadly on developing statistical methods and theory for dealing with kind of advanced biological data, including single cell, and she's been working on Mendelian randomization and more recently also on aggregating p-values to develop powerful methods. And so with that, I please welcome Jingshu.
So thanks a lot for Ahmed's introduction, and thanks everyone for coming, and very honored to have the chance to visit the department. So today I'm going to share our recent work in aggregating dependent p-values. This is joint work with my student Lin at U Chicago Stats, and also my collaborators Yu Chao from Texas A&M, Tian Tian from USTC in China, and also Ruo Du from University of Waterloo. So these are the two papers that are related. If you feel the talk is interesting and want to see more details, you can refer to this. So aggregating p-values has been kind of a long-standing problem, and it appears in different applications. So to set up the context, I want to start my talk with 3 motivating examples so that you can see like what kind of problems we're interested in dealing with here. So the first example comes from spatial transcriptomics. This is a relatively new technology where this technology measures both the gene expression profile and also spatial locations of the cells in the tissue at the same time. And as an illustration, here is the expression of one gene. The height and colors represents the gene expression, and the locations are the 2D locations of the cells in one slice of the tissue. So for this paper, they're interested in finding out genes whose expression change with spatial locations. And they built this generalized linear spatial model to detect such signal. So one challenge they face is that if the gene expression really changes with the spatial location, they actually don't know like what the spatial pattern looks like. So their solution is that they will try many different kernels, and each kernel is powerful in detecting one type of spatial signal. And then they will, each choice of the kernel also results in one p-value. And then they will combine this 10 different p-values to find the signal. So you can see here, combining these p-values actually is not that trivial because the p-values, they come from the same data set. So they might be highly dependent, and it's not easy to characterize their dependent structure. So the second example is in this model-free permutation test. So the authors here, they want to develop some methods that is kind of non-parametric. So their way, and also powerful. So their way is that they will split data into 2. For one half of the data, they will train some machine learning models to find out a very powerful test the statistics, usually coming from a prediction model. Then the second half of the data is used to generate a permutation test p-value. So this is a very flexible framework, but the p-value comes from one random split. So it's random, and also it might be unstable because of the randomness, and it's kind of waste half of the data for testing. So they have proposed in the paper that they will try the random split multiple times. Each time you get a p-value, then you will combine these p-values to get a combined p-value to test for the global norm. And you can see here, these p-values, they come from the same data again, and they can be dependent. And because the model they use is very flexible, it's very hard to characterize the dependent structure. So the third example dates back to this paper about sensitivity analysis in causal inference. So sensitivity analysis is to identify whether there's a causal effect, allowing some level of unmeasured confounding in observational studies. And what test the statistics to use is most powerful depends on the underlying data generating process, which is not 100% settled because some major confounding is allowed. And so what in Paul's paper is that he proposed that you can try multiple different test statistics that is powerful under some type of data generating process. And then you will each test the statistics result in one or several dependent p-values. And then you will combine these p-values to identify the causal effect if any of the tests reject. So you can see in all these 3 examples, you can summarize as their tests. They have like multiple p-values and they want to combine them to test for the global null, which holds when all of the individual nulls are true. And here the number of p-values they want to combine is relatively small. But then the challenge is that these p-values, they may have dependent structure that is very hard to characterize. So for such scenario, one method that might be well known to everyone is the Bonferroni's global test. So it rejects the global null when n times the minimum of the p-value is less than the significance level. And as we may all know, the benefit of this test is that it's valid under any arbitrary dependent structure of the p-values. But at the same time, the drawback is that it can be a very conservative method. It can be more conservative than using any of the individual p-values. For instance, if all the p-values are exactly the same under a complete dependent structure, then Bonferroni's test will reject global null when the individual p-value is less than alpha divided by n. If you use the individual p-value, the cutoff is alpha. So that's actually not very ideal to use in practice. Remember here, our goal is to increase power by combining these p-values. And there has been some improvements like Holm's procedure or generalized means, the generalized Holm's procedure. But in practice, they don't improve. The power improvements over Bonferroni is very minimal. So in recent years, at least within our genetics community, there is an increasingly popular method called Cauchy combination test. Maybe you have heard of it before. And the test has 3 simple steps. So the first is that you choose, like, you use the standard Cauchy distribution. You transform a p-value into using the quantile function, you transform it if the p-value follows uniform distribution on the null, and the transform the statistics follow a standard Cauchy distribution. Then you take a weighted average of this transformed statistics. And the third step, you just compare again with the standard Cauchy distribution. You use the tail probability as your combined p-value. So you can see like this test, why it's so popular. So it's very simple to compute. And you don't need to know the dependent structure. And another thing is that, at least in some special cases, it's very clear that it's more powerful than Bonferroni. For instance, if your p-values are all exactly the same, then if you you can do some calculations yourself, this Cauchy combination test actually does nothing. So it just rejects the global null when the individual p-value is less or equal to alpha. But then if you're using Bonferroni, it rejects at a much lower threshold. So then you may wonder why it can be used. So what validity guarantee does it have? Actually, the original authors that proposed this Cauchy combination test also have shown this important property about such tests, which is called asymptotic validity. So basically, they assume that if you have test statistics that are bivalent normal, and then you also have p-values that are two-sided, then they can show this asymptotic validity in the sense that the empirical type error divided by the nominal type error will converge to 1 when the significance level drops to 0. So this is not exact valid, but practically it's actually typically enough because our significance level is typically very close to 0. And in many genetics applications, actually the cutoff can be even lower, much lower than 0.05. So that's the asymptotic validity they have shown. Okay, so the first time that I get to know this Cauchy combination test, I actually feel it's kind of a magical method. And what is so special about Cauchy? Right. It turns out that there's another test that's also becoming increasingly popular called harmonic mean p-value. And you can understand it through a similar perspective. So the harmonic mean p-value just take the harmonic mean of the n p-values as the combined p-value. Basically, instead of using the Cauchy distribution, you can think of it using the Pareto, standard Pareto distribution. So the density function looks like that. And then, so you first, as the Cauchy combination test, you first transform individual p-value using the quantile function of this Pareto distribution to a Pareto distributed random variable. And then you take a weighted average of this transformed statistics and compare with Pareto again. And then you get exactly this harmonic mean p-value. Okay, so given this setup, then the first natural question is that can we define then a general class of transformations that you can be used in a similar way, and what is special about the Cauchy and Pareto distribution? And the second assumption, second question is about validity. So previously, previous people have shown that the Cauchy combination tests are symptotically valid under for pairwise normal test statistics. So do we have more general like type of error control for this class of combination tests? And also the third question is we are saying we're gaining power over Bonferroni, but like how much power can we really gain and under what scenario? So I'll try to answer these questions during my talk. So any questions so far about the setup? Okay, so let me now continue. So the first thing I want to show you is that actually both the Cauchy and Pareto distribution, they belong to this regularly varying tail distribution subclass, which is subclass of heavy tail distributions. And the tail heaviness is characterized by this gamma, which is called the tail index. And the larger the gamma is, the lighter the tail of the distribution is. And probabilists have shown this property that's very relevant to our context. Actually, they have shown that if you have n random variables that are pairwise symptotically independent, here for symptotic independence, it means that the conditional probability of one random variable being the tail, conditioning on the other random variable being the tail, will converge to 0. So basically, these two random variables, they can be dependent, but at the tail, they kind of look like independent. One example is that if you have joint normal random variables, they're always asymptotically independent as long as they're not perfectly correlated. So if you have n random variables, they're pairwise asymptotically independent, and if these random variables marginally, they're regularly varying tailed distributed, then people have shown that the tail probability of the summation is approximately the same as the summation of the tail probability, regardless of the dependent structure. So it shows some robustness of like this summation against the dependence. Okay, so then like given this class, then we can define this general like class of heavy tail combination test. So we start with p-values, either one-sided or two-sided. You pick one distribution from this regularly varying tail class, and then you can do like a quantile transformation for each p-value to get a transformed statistics. You take a weighted summation of this statistics and compare it with some quantiles of your selected distribution. And that cutoff can depend on both the weights and also the tail index of your of the distribution.
Oh, that's so interesting. So I had a question about this kind of basically almost independent in the tail.
Mm-hmm.
So in the case where it's like heavy tailed, which is like the setting that you're maybe trying to think about, I'm just having a hard time understanding this independence in the tail. So like if I think about extremes, like extremes literature, they characterize dependence in the extremes, like in the tail. In here, you're kind of assuming independence.
So at this moment, yeah, the condition is like your test statistics are pairwise normal.
Yeah.
And then if you do these transformations, actually this transform the statistics, they're independent in detail. And then like under these conditions, we're showing some properties that later.
Pairwise normal.
Yes, that's right. And then you later actually you will see like we have more general considerations, but this is the start. Because I think like people are thinking that practically speaking, having bivariate test statistics with unknown correlations already kind of general, so they start from here.
Yeah, and can you just talk a little bit about that assumption of like the normal, the pair being normal? How reasonable that assumption is? Where does that?
We actually don't know. Like you may think, well, test if like we have asymptotic normality theory, then maybe you can say like if your sample is large enough, then like test the statistics are like multivariate normal, but it's just an assumption.
Mm-hmm.
I think like here we're just for general like conditions. Actually, people are not well. You can set up some conditions, and then here actually the focus of my talk is showing that this assumption can be too restrictive. But then existing papers they always start from here, so that's where I start.
So if we have a sequence of test statistics that are asymptotically normal, generally, of course, right? By multivariate centering theory, can we say anything about they are asymptotically independent?
Depending on which asymptotics, like because here for the first like asymptotic normal, you have sample size go to infinity, yes. And here we also have like the tail go to infinity, and then here it's just I would say like you have these assumptions because it's easy to start at this moment.
But you still have to rely on some asymptotic independence in the finite sample.
Yes, I think at this moment, yes.
Okay. Thank you.
Okay, yeah, and then like you can also get a combined p-value following the same logic. Okay, so here is a class of like heavy regular variant tail distributions. Some common examples: the Cauchy distribution have tail index 1, and then it's like a special case of the student T distribution. The tail index is actually the degree of freedom. And then you can see most of these distributions is actually left bounded except for the student T distribution. I'll show later that if you use this distribution for transformations, having a left bounded support is beneficial. So if you want to make the student T distribution also be left bounded, you can use you can just left truncate it, and it's also within this class. Okay, so this is saying that if you do have this setup, you have test the statistics that are like bivariate normal, and then with no perfect correlations, then like if you use this heavy tail combination test, regardless of what F you choose, you can show that it's a asymptotic value in the sense that the empirical time error divided by the nominal time error will converge to 0 when the significance level drop, or converge to 1 when the significance level drop to 0. And
but in the previous example, you show that even everything is the same, it's still matching the.
That's not satisfying. Yeah, so that's when everything is the same, it's actually not following this assumption because here we don't allow perfect correlation. Actually, we can show that if you do have something that are perfect correlated, we can show that this is less or equal to 1. So that's covered. Everything is the same. That's covered as a special case here.
I may be completely misunderstanding, but when rho ij, every rho ij is 1. Your previous example said it's exactly 1, right? The limit of this fraction is exactly. Well, every rho ij is.
When every rho ij is 1, then
so that's exactly the same.
Yeah, that's that's a special case for the Cauchy. Yeah, so that's a special case where like this ratio is actually exactly 1. Generally, if some of them are exactly 1 or minus 1, we can show it's less or equal. For instance, if it's minus 1, then you can cancel it.
So you can find a special tail -1, so that it is not 1. The limit fraction is not 1.
Yeah.
Oh, so, yeah. Okay, thank you.
Okay, so actually this is not a surprising result because I remember I've shown this probability to show that the summation is not very sensitive to the dependence. And it's slightly, and this result actually slightly sharpens earlier result. Okay, so everything that I've shown you here now does not seem it's reasonable, but may not be that surprising. Okay, now I show you something that might be unexpected. So first, let's look at how the rejection region looks like for this heavy tail combination test. And as an example, this is when n equal to 2. You combine 2 p-values, and I'm showing you in the space of test statistics, we have like 2-sided p-values. And here, the rejection region is the outer space of the boundary. And for the Bonferroni test, it's actually this black square. So first you can compare with the Fisher combination test, which is like using it's also through a transformation, but it's through a light tail transformation. You can see the rejection regions is very different from Bonferroni, and we may like know that the Fisher combination is very sensitive to the dependence of the p-values. And then these are the other two are on the contrast for the heavy tail combination, such as here I show you the Cauchy combination and also the combination using a Frechet distribution. Actually, they look like much similar to the rejection region from a Bonferroni's test. And actually, like if your transformation has like a positive support, we can it's easy to see that for every fixed alpha, it strictly dominates the Bonferroni test. You see there are some like undesirable sharp points. For the Cauchy combination, that's because it has an unbounded support, and that can be corrected if you just like left truncate the Cauchy distribution, use the truncated Cauchy distribution for transformation. So what I actually want to show you that's maybe more interesting is here, if you do look at how the rejection region evolve when your significance level drops to 0, which is the symptotic regime where we show these tests are symptotic valid. Actually, the rejection region looks like more and more similar to Bonferroni, right? So then you're kind of getting worried, like, are we really gaining anything from Bonferroni? Because we only have validity when this significance level drops to 0. Unfortunately, we can show that no, under the same like conditions that people have your risk considered, whether a normal case, there's no like asymptotic difference between this combination test and the Bonferroni test. So specifically, the here what we show that's the non-overlapping rejection region divided by the marginal power, divided by the power actually converges to 0. So in the same asymptotic regime, actually there's nothing different from the Bonferroni's test when you have bivariate test, normal test statistics under like no perfect correlation. And so that's like kind of it's interesting, like even though like this test has been like increasingly popular to use in practice under this setting where people have shown its asymptotic property, actually there's no difference to the Bonferroni. Okay, and this is a kind of empirical like illustration. Here the x-axis is the significance level, so it's the minus log 10, so the significance level actually drops to 0 in the x-axis, and the y-axis is the empirical ratio between the non-overlapping region probability and the power. And it does here we show 3 scenarios under the global null, which is the noun case, and 2 alternatives. One is the dense alternative, the other is the sparse alternative. Because in all 3 cases, actually, this ratio indeed dropped to 0, though sometimes very slowly when the significance level dropped to 0. Okay. So then basically what I'm showing you here is that seems that theoretically there's no difference between Bonferroni's test and this fancy heavy tail combination test, though empirically it's used like very commonly. But then we also realize that's not the full picture of the story. So remember, like when I introduce the motivation of this test, I consider this special case where all the p-values are exactly the same. And I'm saying that when all the p-values are exactly the same, Bonferroni's test rejects when p-value is less or equal to alpha divided by 10n, so it's very conservative. But then if you use this heavy tail combination test, it rejects when the p-value is less or equal to alpha. So that gap is actually not diminishing when your significance level drop to 0. So what is happening here? So actually, if you compare these 2 cases, the first is under asymptotic dependence, and the second is when like these p-values are completely dependent with each other. So then that motivates us to think maybe we should consider a broader class of dependent structures when we're trying to understand the validity and power of these combinations tests. Maybe these tests are still valid and they're more powerful than the Bonferroni's test. And in terms of validity, I think Fang and Emma has already questioned, I think, like this asymptotic independence is actually a weak guarantee because validity, we only show validity when the significance level drop to 0. But then we required that these p-values, they appear independent at the tail. So that's not really a strong guarantee of validity when our goal here is actually kind of, we try to apply these methods in real applications where we don't know the dependence.
That's good. So I guess like in some sense, Bonferroni, when you have independent p-values, Bonferroni is in some sense tight because
yes,
it like the approach is alpha, and this is kind of the message you're trying to say here, right? Like,
yes, even though like they are dependent, but if they're independent in the tail, it's also very hard. It's not able to be bound. Yes, that's right. So we just want to make this method more concrete and showing you like what type of dependence.
A remark is any possible gain is under assumptions, and
yes,
these assumptions may be wrong, and the test may even not have a set control if the assumptions are wrong. But Bonferroni is always correct.
Yes, that's right. So
that's
like
the beauty of Bonferroni.
yeah, that's right. So if you want to gain over Bonferroni without any additional assumptions, then you can get very, very minimal gain. But then like we're trying to find like some assumptions we think that are like very likely to be possible in practice, and then you have some gain over Bonferroni. So they kind of safely use these approaches that are more powerful for you to help you discover more signals. Okay, I'll show you what's the assumptions that we really have. Okay, so this is an empirical comparison. So here I'm showing you, so here I have n equal to 5, 20, and 100. And here the x-axis are the dependence, the pairwise correlation. So here the significance level is controlled at 5 times 10 to the minus 4. And the y-axis is when you increase signal from 0 to infinity, what is the maximum power gain over Bonferroni, power difference. Okay, and then like this is what you have when the test statistics follow multivariate Gaussian distribution. So what I want to show you is a contrast that if I just making everything the same, but I just change the test statistics from following multivariate Gaussian distribution to following multivariate T distribution so that they are more dependent in the tail, then you can see like the power gain over Bonferroni can be dramatically increased.
So maybe this is a question that is better for the end, but let me ask it now because maybe you can address it as you go. So I'm familiar with kind of this literature on extreme values from like more of the environmental perspective, right? So where, for example, you think about max stable processing as a way to have tail dependence, which is in some sense the opposite of what you're getting here. But I mean, but if you have things that follow multivariate T, you're going to have tail dependence as opposed to the multivariate normal, which means that if you have correlation in space, like for example in the phase transcriptomic case, what you want is a max stable process, giving you the copula for this multiple P values that you're going to transform. You want to transform with the joint copula. So I'm having kind of a bit of a hard time thinking about how this connects with the literature on dependence in the tail of process or multivariate.
So here we're actually talking about dependence of the p-values. So you may think like there can be like 2 ways of generating the p-values, so that when there's like these 2 different, say like you apply 2 different tests to generate p-values, and these tests maybe they look different, but they are like on the same data. So actually like these p-values, when they're small, they're kind of going together with each other because they're just 2 different methods applied on the same data set. So you indeed may have these p-values actually have possibly are very dependent at the tail. So that is what we are assuming here. Like for instance, if your sample size is not large, then your test statistics will follow multivariate T distribution. Then the p-values they generated will also have high tail dependence.
This is unclear whether like the max stable distributions arise kind of naturally. It'd be kind of interesting whether there is a setting where the p-values naturally will have them.
Well, I mean, this is the opposite situation, right? Which you're saying, I want tail dependence.
Yeah.
And it contains the tail independent special case, if you want. And so So the question is, if you assume this max table type distribution for the multiple p-values, like what do you really need? So in that space, do you really need independence, or can you have certain settings of a max table process where there is some level of dependence built in, and you still have some of the theoretical gains that you're having?
So
that's the other thing.
So I think I will show later. Actually, we have like more simpler definitions, so that it look more natural for like when you talk about copulus of the p-values. It does have connections with the max available thing that you talk about, but let's see later. Okay. Okay, so now before I show you what results we really have, let me first summarize our theoretical results informally, intuitively. So first, like as I said, we're considering a larger class of dependent structure that include both asymptotic independence and the complete dependent special cases. And it's a class of copula where we call the multivariate regularly varying copula. And then, like you will see later, actually, it's actually a very large, broad class of dependent structure. And then what we can show is that for this heavy tail combination test under the MRV copula, these tests are asymptotic valid if the tail index of the transformation distribution is less or equal to 1. And then, moreover, we actually can show that the power of this heavy tail combination test monotonically increases with gamma, and under lower tail dependence of the p-values, and it's strictly monotonically increasing on the lower tail dependence of the p-values, and when signals are not too sparse. So basically, if you consider like this is a line of gamma, we can show that when gamma is, so remember, like the smaller the gamma is, the more heavy tailed the transformation distribution is. So we can see like the validity, the time error in when the gamma less or equal to 1 is valid, and then the power monotonically increases with gamma. So gamma equal to 1 should be the optimal choice of gamma for the heavy tail combination test. And then how do you compare with Bonferroni? We actually compare. You will see later that the Bonferroni can be considered as a special case of this heavy tail combination test, where you use the most heavy tailed distribution for transformation for like gamma equal to 0. So you can consider Bonferroni test as a special case with gamma equal to 0. And then, like as I said, power monotonically increases with gamma. And more specifically, if we compare the power ratio between this optimal heavy tail combination test with gamma equal to 1 divided by the power of this Bonferroni test, then we can show this power ratio is monotonically increasing with the dependent structure. So when the p-values are symptotic independent, that's where the ratio is 1. So that's like where you have the least power gain. But then your power gain can monotonically increase when the p-values are more and more dependent at the tail, and it reaches the maximum when they're completely the same. So that's the series of theoretical results that we have, and that's hope that will give I think it's give us a clearer understanding of what this heavy tail combination tests are really doing. Okay, so let me show you more formal results first. So let me first define this multivariate regularly varying copula. I think that's related to the earlier question. So here, well, the copula are commonly used to describe dependency structure of a random vector without considering the marginal distribution. And to define this multivariate regularly varying copula, we first define this upper tail dependence function of a copula. Why we care about upper tail dependence? That's just to follow the literature of extreme value theory. And then we say like a copula is an MRV copula as long as this upper tail dependence function has a linear scaling limit. So basically, we're only recovering the existence of a limit, and we only consider this limit in the upper tail dependence function. So any copula, so the other features of the copula actually is not required. And because we only require existence of a limit, it should be a general class of copula. And as an example of how you understand, So this L11 is actually a 2 minus the conditional tail probability of one random variable given the other random variable in the tail. So we kind of only require existence of such conditional probabilities for like for the dependency structure of the random variables. And how we use this MRV copula to describe the dependent structure of the p-value. So basically have some assumptions on the dependence of these p-values when they're near 0. So we are basically assuming that 1 minus these p-values, it follows an MRV copula. So basically we're assuming that this lower tails, when the p-values are near 0, it has this kind of linear scaling limit. So that is the assumption of what copula belongs to this MRV copula. So this is a little bit abstract at this moment to convince you more on like why we think this is a general class. So let me give you some examples. So first, any copula of that are asymptotic independent belongs to this MRV copula. Basically, we only require that they're pairwise asymptotic independent. So the conditional probability pairwise is 0. So that includes the Bavarian normal case as a special case. And any copula of continuous random variable vectors that is asymptotic complete dependent is also a special case of this MRV copula. And we also then can see most of the known copulas are belonging to this class. So one example is the Gaussian copula, and the multivariate T copula also belongs to this class. And many survival Archimedean copulas also belong to this class. For instance, the Clayton copula also satisfies the MRV copula. And the extreme value copula is another special case of this MRV copulas. And also, if you have mixtures of this MRV copula, it's a convex. So any mixture of the MRV copula is still an MRV copula. So what I want to show you is that if you want to, so here we're talking about dependence of the p-values, right? You may want to, so how do you model the dependence of p-values? If you are using some copulas that are having like a closed form formula, then actually it's easy to check whether it has such scaling limit. And actually most of the copulas that has a closed form formula that satisfy, they have a limiting such limit.
So you're mostly talking about pairwise dependence. So if I think about suppose that there is conditional independence between the p-values, that doesn't really matter in this case, because as long as you have high pairwise dependence, you can show power improvement and so on. But if you have if I think about actually there is a lot of structure here that even though there may be a lot of pairwise dependency, there's a lot of conditional independencies and so on. That doesn't really matter at all for sort of results here.
In terms of conditional independence, I think here we're mostly actually if you have multiple random variables, you I think as long as pairwise they're not yes. Because we are saying it's equivalent to Bonferroni when for every pair they're kind of independent, then it's equivalent. So that's the if and only if condition for they're the same as Bonferroni.
And you're also implicitly assuming that the dependence amount is the same for all pairwise?
No, we just so here we're considering this MRV class, which is it only requires that there is a limit. Here just for this these examples, the pairwise is the same, but you can generate cases that are not. For instance, if you have Gaussian multivariate T copula, it depends on the covariance matrix. They don't need to be symmetric. Okay, so here, for the validity results, let's just consider the talk. Let's just consider we have equal weights, and then the transformation distribution is left bounded. So we only consider left truncated Cauchy or T, which has shown to be more powerful than the original Cauchy combination test. And then here we are defining the limiting scale type and arrow. So specifically, we can show that this limiting scale type error is a monotone. It only depends on the tail index of f. And also, more importantly, this Q gamma is a monotone, the increasing function of gamma. So the more light-tailed of the transformation distribution, the larger the Q gamma is. And more importantly, this monotonicity holds is strict if the equality holds if and only if the p-values are asymptotically independent. And Q1 is always 1. So because of this monotonicity, then Q gamma is less or equal to 1 for any gamma is smaller than 1. So that's why, like, for this heavy tail combination tests are always asymptotically valid as long as gamma is less or equal to 1. And we can also show a bound on Q gamma. So basically, this Q gamma is bounded between 1 and n to the power of gamma minus 1, where here 1 comes from like this Q gamma when the p-values are somehow independent. And this n to the power of gamma minus 1 comes from when the p-values are like completely dependent. And here are some simulation results to show you the empirical type of error. So here I'm showing you the transformation with gamma equal to 0.3, 0.6, 1, 1.2, and then the dotted lines are the boundaries for n to the power of gamma minus 1. You can see the empirical power well like kind of between these two bounds, and it kind of also monotonely changes with the dependent structure. And here you can see like when alpha equal to 0.05, there was a regional test with gamma equal to 1 slightly, like not controlling the empirical type of errors. If you really care about in practice, you can kind of adjust it a little bit on the independence. So this is what is the empirical power validity look at when n equal to 100. So here I'm just trying to show you that is theoretical, sorry.
So to implement your test, you need gamma. You need to know the gamma.
So gamma is the transformation distribution you choose.
Gamma is in the multivariate regular varying calculus.
No, the calcula, we only require the calcula to belong to this MRV class. And the gamma is the transformation distribution that you use to transform the p-value.
Oh, okay, I may misunderstand. So I have a problem.
Yeah, that's right. So the transformation depends on gamma. And here for the p-values, we just assume that the lower tail dependence has some limit. So that's the setup. So the gamma is something we can control. I'm showing you that we need to choose some gamma less or equal to 1. And then we can also define this limiting. Yeah, you have gamma. When you choose gamma equal to 1.2, you can see it's not controlling the type error. Yeah, so we want this to be less or equal to 1, at least in the limit. Then it's shown that when you choose gamma larger than 1, then it's bounded between 1 and some upper bound. So it's not really valid. Okay, so this is the empirical power, and then we can show that this empirical limiting scale power, this limiting scale power also is monotonely depending on gamma. And this inequality is strict if and only if there are at least the 2 dominating signals. I'll explain in the next slide. So intuitively it means like I should have at least the 2 signals that are comparably strong to beat Bonferroni, otherwise not. And also these p-values are not asymptotically independent. So as long as this, then I have like Q gamma monotonely strictly monotonely increasing with gamma. And then that's showing, combining with the previous result, that's showing that the optimal test, we need to choose gamma equal to 1. And this is like the exact mathematical definition of what we mean by dominating signals. Basically, it means that the p-value's tail is dominating. It's not dominated by others, so then that's a dominating signal. We need to have 2, at least the 2 of them. And we also have results comparing with Bonferroni. So basically, we're showing that the power ratio between this any heavy tail combination test and the Bonferroni test is at least larger or equal to 1. So this means that the asymptotic power of the heavy tail combination tests are at least as powerful as Bonferroni. And it's strictly better than Bonferroni when you have at least have 2 strong signals that are comparable with each other. And the other is that these p-values are not asymptotic independent. So if you have both these conditions, so this is like a sufficient and necessary condition for it to strictly beat Bonferroni. And also Bonferroni can be considered as the limiting case of this heavy tail combination test with gamma equal to 0. Remember, we have shown that the power actually monotonely increases with gamma. So gamma is the least powerful heavy tail combination test, and gamma equal to 1 is the most powerful heavy tail combination test. And they are equivalent when things are asymptotic independent. And specifically, we can show that if you look at this power ratio between the most powerful heavy tail combination test and the Bonferroni, which is the least powerful heavy tail combination test, then this power ratio is monotonely increasing with dependence under some partial order of the dependent structure. And this partial order is like, so there is equivalent when they are asymptotically independent. The power ratio is maximized when things are completely dependent with each other. So in other words, these Bonferroni tests are increasingly powerless with increasing dependence among the p-values. I think that's intuitively everyone believes that. Here is just a more formal characterization of the properties of Bonferroni. So here is the formal definition of what order we have to describe dependence. So basically, you need to, for this MRV copula, you need to define its limiting copula, and that characterizes dependent structure in detail. So for every MRV copula, there's a limiting copula. If the copula is asymptotic independence, then the limiting copula is just the independence copula. And if the MRV copula are asymptotic complete dependence, then the limiting copula is like complete dependence copula. And then this pointwise order is just saying like the limiting copula has this pointwise order. And as an example, for instance, for 2 T copulas, it follows this order if the correlation matrix are pointwisely like one smaller than the other. And we have this result that for any like MRV copula is actually bounded by the asymptotic dependence copula and the asymptotic copula. So okay, and then like this is an empirical illustration of the power ratio. You can see like here, so x-axis is again the dependence level, and this is for n equal to 100. So this is the power ratio between the optimal combination test and the Bonferroni on the dense signal. You can see this power ratio, well, it can reach like 1,000 times. That's why it's such a high power ratio. So this is another figure illustrating how the power changes with signal levels. So you can see that in this dense signal scenario, you may have like Bonferroni still around power of 0, but this red curve, which is the optimal heavy tail combination test, already reach a power of 1. So that can be the difference that they can have. Okay, I have maybe 1-2 minutes, and I'll show you some real data examples to give you an idea of how it works in practice, and that also shows why it's like kind of practically very popular. So this is the first example is on like a gene-based association testing, genome-wide association studies. So here for each gene, we're combining the genetic variants within the gene region. So for each gene, we have like 20 to 200 p-values to combine. And then we need to do a multiple testing correction across all the genes. That's why I mentioned the essential significance level for each gene actually can be much less than 10 to the minus 0.05 because of the multiple testing correction. And here I'm showing you the number of genes that are rejected. Okay, so when FDR is at the level of 0.05, like you can see, these are different methods. The first two are like domain-specific methods where they try to calculate the dependent structure and design a test that's powerful. And so these are just using the generic like heavy tail combination test. This is using Bonferroni. It rejects about 400 genes. And then if you're using this, this is actually the truncated Cauchy test, and they can reject 500 genes. So that's actually 25% more powerful, more genes than using Bonferroni. And if you have like a smaller cutoff, like if you have a more like a generous cutoff, then actually it can reject 50% more genes than Bonferroni in practice. So the second example is in circadian rhythm detection. So here is circadian rhythm is the like inner biological clock of our body that corresponds to the sunlight. And here we're trying to find out which genes are controlling this process. For each gene, we need to combine about 200 p-values that are like we calculate at different phases and periods. And here we have a set of negative control genes and positive control genes. So for the negative control genes, you can see because this p-values are dependent, if you're using Fisher, it's actually very wrong. If you're using like this heavy tail combination test, they're not uniformly distributed. But remember, like we are rejecting at some cutoff. So you can see like we actually cannot reject anything for the negative controls. And then for the positive controls, it's more powerful than Bonferroni. So quickly, this is kind of a third example, and we use it to combine different methods on one data set. I don't have time, but basically this is each dot is a data set, and we're combining 5 different methods, the p-value from 5 different methods. You can see these are the number of rejections if I'm using single methods, and these are like using these combination methods, we actually can have a power gain over individual methods. And actually here it performs similar to Bonferroni because like most of the in practice, like some turns out typically a single p-value dominates other p-values. Okay, so I don't have much time, so that's let me just end here. So if you feel interested, you can look at these papers for more details. Thank you.
We have time for 3 questions. I have 4. For the FDR part, so I guess we're in a world where we have dependence, and I have this question of is it easy to incorporate like PRDS, like conditions of like positivity type conditions that you need for I don't know what you're using for FDR, but like for using something like Benjamin Hodgberg to incorporate that in the models, in the copula models that you are.
Actually, we here, so here the setup is slightly different because here, actually, for each gene, we have to combine p-values to test for global null.
Yeah.
And then in previous results are saying that then you have some kind of, there are asymptotic valid p-values. And then you just combine these asymptotic valid p-values using BH. Sorry. Then you're just doing multiple testing across these asymptotically valid combined p-values. As you are just
for the multiple testing to be valid, don't you need? Wait, but here is asymptotically dependent, right? Because in the tails you are dependent. I'm a bit confused, I guess.
So you are first combining within each gene. So that's across genes you need like regular assumptions that you normally. So yeah, so here it's just like it's 2 steps. So previously I'm just talking. So the previous step appear in the first step where you are combining the SNPs like within the same gene. You are using it's a global test and they are dependent. You use this heavy tail combination test, and then you are doing multiple testing correction across the genes. Then that's a different thing.
Thanks. Any other questions? Maybe so. Thanks to the speaker again.
Thank you.
I guess I'm still wondering if I had to build in the positive dependence in these p-values, even let's say across the genes that there was some tail dependence. And
so you are talking about build PIDRs when I'm combining p-values?
No, like across the genes, you assume now like, you know, you have PRDS or whatever, right? But let's say,
Yes, of the combined p-value.
yeah, but let's say you have tail dependence still across the different genes, and you want to build in, you want to do similar things as you were doing with the copulas, but across genes, and you wanted to build in the PRDS condition. How do you, is it possible to kind of model those?
Yeah, I think these are different assumptions. From what I understand, they say for the pH, I can also maybe, or I don't know, say if I'm applying a closure principle across this gene, then for each combination, I can also do the heavy tail combination test,
That's
and
right.
that needs like this tail dependence, and it's actually different from the PRDS assumption. I think here, I've not considered it very carefully, but I think these are different types of assumptions. So here we are not caring about the dependence, not at the tail.
Yeah.
And we don't just allow, well, I think it has some relationship because actually this here, the transformed, well, the original p-values, they can be negative dependent. But you
The
use
combined one shouldn't be somehow.
Yeah, because you kind of use a left truncated distribution
Yeah.
for transformation. So even the original p-values, they're negatively dependent with each other.